I'm Tia,
I'm Jacqueline.
and this is Chapter 3, The Cell, the Living
Cytoplasm. It contains everything in the cell. Cytosol is the juice in the cell that provides liquid environment for chemical reactions to take place. The organelles in the cell include mitochondria, which are these. They are responsible for ATP synthesis. These are ribosomes. These are the free ribosomes, and these are membrane-bound ribosomes. This is the rough endoplasmic reticulum. It is the site for protein synthesis. This is the smooth endoplasmic reticulum, and it is responsible for lipid synthesis. This is the Golgi apparatus. It is also easier known as the UPS of the cell. It modifies, packages, and transports cellular material, but don't call it the UPS. These are the centrioles. They are responsible for creating the mitotic spindle. Not pictured in this model are microvilli, which increase surface area. They are extensions from the plasma membrane. Also not pictured are lysosomes and peroxisomes. Lysosomes dissolve cellular material. Peroxisome detoxifies cellular material. Now let's take a closer look at the nucleus. The nuclear envelope is a double phospholipid bilayer. It has little pores to let RNA out of the nucleus. Nucleoplasm is the cytoplasm inside of the nucleus. Nucleoli, plural, 1 and 2, they make ribosomes. Nucleolus, singular. Chromatin is non-condensed DNA.
Here we have membrane lipids, which consist of phospholipids and cholesterol, and they make up the lipid bilayer of the cell membrane. More specifically, this phospholipid bilayer lies on the inner and outer layer of the plasma membrane. These phospholipids consist of a polar head and a nonpolar tail. The polar head is also hydrophilic, meaning that it loves water. Therefore, it is on the outer layer of the membrane. The nonpolar tails of the phospholipid bilayer are hydrophobic, meaning they despise water. This phospholipid bilayer is what makes the plasma membrane semi-permeable by only allowing certain materials to pass in and out of the cell. In this phospholipid bilayer, we also have cholesterol, which is a 4-ring steroid structure that makes up 20% of membrane lipids. It also helps to stabilize the membrane and decrease solubility in the membrane.
So here we have membrane proteins. Transmembrane proteins go all the way through the membrane. Integral proteins only go through one side of the membrane, and peripheral stay on the outside of the membrane. It is important to note that transmembrane proteins are responsible for channels and carriers.
Here we have our 3 types of membrane junctions: tight junctions, anchoring junctions or decisomes, and we also have gap junctions. Tight junctions is when membrane proteins fuse together to create an impermeable barrier to prevent the movement of materials. Anchoring junctions or decisomes hold cells together with a strong grip and are good at resisting stress. Finally, gap junctions. These allow cells to communicate with each other by only allowing certain materials in and out of the cell, demonstrating that the plasma membrane is semi-permeable.
So now we're talking about movement of materials through the plasma membrane. There's 2 major kinds. First being passive, which does not require energy. It is also known as simple diffusion, which allows things to flow freely in and out of the cell. Passive also moves down a concentration gradient. A concentration gradient goes from a higher concentration to a lower concentration. There's also, in the category of passive, is facilitated diffusion, which has carriers or channels. The third is osmosis, which is the diffusion of specifically water across a semi-permeable membrane. In osmosis, there's two types of pressure. You have hydrostatic pressure, which is the pressure of water, hydro, and you also have osmotic pressure. This is the pressure of salts. All right, we have three types of solution. Isotonic, the sodium in water is equal, so the red blood cell stays the same. In hypertonic solution, there's more sodium outside of the cell than there is inside, making it a hypertonic solution. The cell shrinks and this is called crenation. In a hypotonic solution, there's more sodium inside the cell, which causes the cell to blow up. This is called plasmolysis.
So continuing on with the movement of materials, we already talked about passive transport, so now we have active transport. Active transport requires energy because it moves up or against the concentration gradient. This is done through vesicles, which are membrane-bound bags of cellular material. The two types of vesicular transport are endocytosis and exocytosis. So we have exocytosis and endocytosis. Exocytosis is the process of moving materials outside of the cell, and endocytosis is the movement of materials into the cell. This is done through phagocytosis, pinocytosis, and receptor-mediated endocytosis. Phagocytosis is when the cell eats or engulfs materials to dispose of. Pinocytosis, or cell drinking, is when the cell absorbs a small amount of liquid. And finally, receptor-mediated endocytosis. The cell uses receptors to catch molecules that the cell wants.
Unlike most of us, the cell has a life. Before mitosis, we have interphase. There are three stages of interphase. G1 is the cell living its normal life, having a grand old time. The second phase is synthesis. This is where the second copy of DNA is made. And last, we have G2, which makes specific proteins for the division process. These are the stages of mitosis. First, we start with prophase. Prophase, chromatin condenses into chromosomes, nuclear envelope breaks down, and the mitotic spindle forms. In metaphase, the chromosomes line up at the equator of the cell, creating the metaphase plate. The third stage is anaphase, where chromosomes move away from the metaphase plate. In telophase, chromosomes stop moving, new nuclear envelope forms, chromosomes turn back to chromatin, and the mitotic spindle disappears. If you need help remembering the order of mitosis, think PMAT.
Protein synthesis, or gene action, is the making of new proteins using RNA, or ribonucleic acid. Here we have three types of RNA. We have mRNA, or messenger RNA, which is the recipe for protein. mRNA determines the order of the amino acids and puts the primary structure in order. We also have tRNA, or transfer RNA. This brings the appropriate amino acids to the ribosomes. It brings in the raw materials. And finally, we have rRNA, or ribosomal RNA. It is composed of ribosomes. So in protein synthesis, the role of RNA is carried out in two steps: transcription and translation. Transcription is making an mRNA copy of DNA. Translation then turns this copy into a protein using mRNA, tRNA, and rRNA.